So the first part of the lesson today, we're going to look at these metabolic reactions. What is meant by metabolic reaction? So generally, metabolic reaction is a set of life-sustaining chemical transformations within the cells of the living organism. Okay, and this metabolic pathway begins with a specific molecule, and then we alter them in a series of the reaction or defined step, result in a certain formation of the certain products. And the metabolism is usually divided into these two categories, which are catabolism or catabolic reactions, and anabolism, which is anabolic reactions. When you look at the catabolism, basically catabolism is a metabolic pathway. At the end of the process, you're going to release the energy by breaking down the large molecule into the smaller molecule. So example of catabolism that always happen in our body include the cellular respiration, where we break down the glucose molecule, okay, by oxidations in the presence of oxygens to carbon dioxide. Okay, so we do the oxidation process to our glucose molecule and energy is released. Okay, how about anabolism? Anabolism basically is a pathway that require energy or ATP so that we build a large molecule from a small compound. Okay, so example of the anabolism that we do know, like for example, synthesis of protein from the amino acid, synthesis of the glycogen from the glucose, okay, alpha glucose, and also synthesis of the, for example, cellulose from beta glucose, synthesis of the mRNA, or anabolic reactions. Okay, so students always confused. We do know that to break the bonds, we need to absorb the energy. Correct? In chemistry, you learn this, right? Okay, to form the bond, release the energy. But why we say that breaking down a large molecule release energy, but when you build up the molecule, we require energy. So this, we have to know that for any reaction take place, they involve both bond breaking and bond forming. So this part will be slightly, how to say that? The concept must be correct first before you can actually understand this part. Okay, so let me show you what is the concept. In chemistry, you always remember it's not wrong that what you learn is not wrong, eh? Because we have to understand that when you break the bond, chemical bond, then definitely, you need to absorb energy. Okay? Absorption of energy. Energy absorption take place, right? Just now we say that we want to break the hydrogen bond, we need to absorb the energy. Okay, and for the bond formation, when you form bond, then energy is released. Okay, so but in the catabolic reaction versus anabolic reaction, what we do here is we not break down the bond only, we also form the bond. So in this case, understand the concept here. Catabolic reaction, actually, we break down macromolecule or large molecule to a smaller one. Okay, we are not talking the bond here yet. Okay, catabolic reaction, we break down the large molecule to small molecule. Okay, anabolic reaction is opposite. Basically, we are going to build large molecule. Okay, I are from small molecule. Are you clear? So understand the catabolic and anabolic reactions now. Now, no matter which one, catabolic or anabolic, it must involve breaking the bond first and then form the bond. It must involve both. You have to break the bond and form the bond. I use a very simple, okay, reaction here, then you understand. For example, hydrogenation on the ethylene. Right? This is ethene, right? So in order to change the ethene to ethane, we can do the hydrogenation. Means that we add hydrogen gas. So try to imagine now. In these reactions, when you add the hydrogen gas, you realize that now I need to break the bond first. If I didn't break the bonds, how can I separate the hydrogen atom? Cannot. So first step, we need to break the bond. Cannot. And then we form the new bond. So in this case, when you break the bond, then you absorb energy. Heat energy, for example. Cannot. And then when you form back the bond, you release energy. So now it depends during the process whether absorbed energy is higher or released energy higher. So if absorb more than release, these reactions is called endothermic. Are you clear? If we absorb more energy than release, so means that there is a net absorption. Cannot. Let's say we absorb 10 units, release 5 units. So means that overall we still absorb 5 units. Can you see that? For example, I absorb 10 units, release 5 units. So overall, I still have net absorptions of 5 units of energy. So therefore, this process is termed as endothermic reaction. So endothermic reaction generally applies to all anabolic reaction. Okay, generally, anabolic reactions are endothermic reaction. Are you clear? So means that if I draw the diagrams, okay, so this is the energy level. This is the time taken, okay, for the process to take place. Then in this case, the reactant have low energy compared to the product. Okay, products, reactant. So reactant have low energy, so that's why they absorb the energy. Clear now? Okay, so therefore products will have more. So this case, you can see that this is a change in the energy. So the change in energy in biology, we call it a delta G. So delta G is positive. So means that is the endothermic reaction. So it's the energy requiring process for anabolic reaction because absorption, the energy needed, okay, to break the bond, okay, is more than the formations of the bond, the energy release. Are you clear? Okay. So for all chemical reactions, then you can see that it's not straight away, the energy required is not straight away up to the these products. You have to go through a loop like this. Are you clear? Go through a loop. So when you go through a loop like this, so this one we call it as a transition stage. So for reactant to reach a transition state, this is termed as activation energy or EA. Okay, it's termed as the activation energy. Are you clear? Okay, so now in opposite, if let's say energy release more than the energy absorbed. Okay, so this process is termed as the exothermic release energy, right? So exothermic reaction, okay? Exothermic reactions. So that is a net release in this case. Okay, sorry. Let's say, for example, I release 15 and absorb 5. So that is a net release of 10 units of energy, the net release. Can you see that? So this kind of reaction generally applied to the catabolic reaction. Okay, so if we draw the energy diagram, say, this is the energy, this is the times taken, then you can see that reactants having the higher energy compared to the product. Okay, so the difference in the energy here is termed as the delta G also, but this delta G is negative, indicate that it's exothermic reaction. Are you clear? It's exothermic reaction. But again, you can see that we have to overcome what we call the transition stage first before we can get into the product. So this is a transition state. So in order to overcome this, you have this amount of energy you have to overcome. This amount of energy is termed as activation energy. Are you clear? Okay, so make it smaller. If you need to copy it out or you need to take the photo, you can. So in these two diagrams, you can see that for the reactant to become products, they have to go through this transition state. And this is very, very important. Most of the biochemical reaction in our body or living organism, activation energy is very, very high. Now, why? Why any activation energy is very, very high? Activation energy is very, very high in our bodies so that we can control the metabolic reaction. Now, again, let us look at this energy level again. Okay, so this is the activation energy that we need to overcome. Are you clear? Okay, so now how we overcome the activation energy? Now this activation energy exists here. Why? Because for two particle, okay, to react, let's say we have particle A and particle B to react. First thing, they need to collide on each other, right? They have to collide on each other. When they collide, they have to collide correctly. Okay, if it collide wrongly, the bond cannot be formed and cannot be broken. So this activation energy. So one of the way we overcome this activation energy to make sure that more effective collision take place. Let's say, for example, this is A. This is B. Okay, so the correct collision is this orientation. But if they collide this way, so no reaction. This one reaction take place, no reaction take place. Are you clear? So this kind of collisions is termed as effective collision. Are you clear? This kind of effective collision is effective collision. So these effective collisions allows the bond formation and the bond breaking, forming a new product. Okay, so energy required for this reactant to go to this transition stage is termed as activation energy. So in order to have more effective collision, one of the common way we do it, we increase the heat energy or increase the temperature by supplying the heat energy. So a lot of reaction why we boil it, right? Why Benedict reagents, okay, Benedict test or this reagent sugar test, why we heat in boiling water bath? Because we want to have more effective collision. Means that when you supply the heat energy, kinetic energy of the particle or reactant is high, so they collide even more. So increase the chance for the collisions, effective collision to take place. Not the normal collisions, but effective collision to take place. Clear? But the thing here is for living organism, we cannot place ourselves, definitely, okay? We cannot throw ourselves into the hot water bath. Oh, I want this reaction to take place. I go in, put myself into this hot spring, for example. And if you can heat, let's say, for example, there's no problem of the denaturation. That is, let's say, if you also got a problem because if I put myself in a hot environment or put myself in this hot spring, then every single reaction will take place. Cannot, we lose the control. So how we can control the metabolic reaction? We do control it by using the enzyme. So enzyme have two functions here. When talking about enzyme, definitely we deal it with the specific. Okay. The enzyme, they are specific. The specificity of the enzyme. So means that only allow a particular reaction to take place. Okay. Second, how do you do this? Enzyme actually lower down the activation's energy. Are you clear? Overall, we can see that it's something like this. Okay. I always say like this. If I tell you that, okay, tomorrow we're going to have class tests, the passing mark is 95 marks out of 100. Oh, passing mark so high. How many will pass? Correct or not? Are you clear? So it means that maybe out of 100 students in the class, only 2 or 3 pass. So this is something like activation energy. Activation energy is so high. Okay, activation energy is so high until most of the reactant cannot react. So very, very small number of the products that can be formed. But now, with the presence of the enzyme, something like tell you, okay, now passing mark is 5 marks out of 100. So majority of the students, even all the students can actually pass this. So means that enzyme lower down the activation energy. So when enzyme lower down the activation energy, so means that more reactant actually can overcome the transition state and form product. So enzyme speed up the reactions, kind of make the process faster. Okay. So now, in first chapter, we always say that nucleus control cellular activity. So this cellular activity actually is the metabolic reaction. Because of this metabolic reaction, then we have the cellular activity, right? For example, detoxification. We also say, oh, liver cell carrier detoxification. How? And how nucleus actually controls cellular activity? Now link. Nucleus. In the nucleus, we have the DNA. DNA have the gene. The gene code for the mRNA. In the transcription process, we call mRNA. mRNA is translated into the polypeptide, where involve ribosome. And then this polypeptide fold, let's say in this case, in the RAR, it fold into the protein. Okay, so this one folding process take place or modification take place in the RAR. Can you see that? Are you clear? And these proteins can become the enzyme. Can you see that? So means that this is how nucleus actually control the cellular activity. If let's say I want a particular process to take place, then nucleus say that, okay, we code for enzyme. We give enzyme. When we give enzyme, then the process can take place, right? So this is how the nucleus actually control the enzyme. Oh, I don't want to have this reaction take place. What are you going to do? Send someone to destroy the enzyme. So without the enzyme, activation energy is high. So this is how nucleus do. Nucleus actually, okay, help us to code for with the DNA, they can code for the enzyme. With the presence of the enzyme lower down the activation energy. So therefore, a particular reactions can take place. And then I don't want to have the reaction take place. What we're going to do? We're going to stop the process. Are you clear? And how nucleus know? How the DNA know? basically they involve the cell signaling pathway, where we're going to learn after the end of these topics. So if you understand the concept now, you can see that these 4 chapters that we have learned, they are linked. First, nucleus, ribosome, RAR, all actually belongs to the first chapter where we talk about the structure of the cells. Second, we learn about the enzyme, the protein, globular proteins, polypeptides, how we fall into the precise 3-dimensional shape. That will be our biological molecule. When it fall to the enzyme, how enzyme carry out the reaction. This will be the third topic. And this is today's topic. We're going to learn the enzyme, the globular shape of the enzyme, the folding of the polypeptide into the precise 3-dimensional shape, allow the enzyme to carry out its activity. And how nucleus know. Okay, how nucleus know? Oh, it's time for me to ask sometimes some enzyme to function. So involve the cell signaling pathway, which is from the chapter number 2. Can I see that? Are you clear? No, haven't done yet. How this mRNA can be formed, how this polypeptide can be formed, we're going to learn in the protein synthesis. So always understand that biology, they are linked. It cannot be topical. So one chapter, one part of the topics you are not understand, it may affect other topics. So I always ask you guys to ask me the question, because if you cannot clarify some parts of the topics, you may not be able to proceed to the next parts of the topics because they are linked. Okay, So we know that every catabolic and anabolic reaction involves the bond breaking and bond forming. So changing one molecule into another generate involve contorting. Okay, we have to contort the starting molecule or the reactant into the highly unstable stage called transition state before the reaction can proceed. And this state to overcome, okay, the energy required to contort, okay, the reactant molecule so that the bond can be broken is known as the activation energy. Okay, so in this diagram, what you need to do now indicates, okay, the activation energy EA and the energy change, delta G, on figures 4.1, and state whether it's exothermic or endothermic reactions. Okay, so again, guys, very simple. Reactant to product, the difference in energy level is termed as delta G. In this case, negative delta G because it's exothermic reactions. So from reactant to reach the transition state, they have to overcome the activation's energy. So same goes to these endothermic reactions. The reactant have to overcome these transitions, the activation energy in order to reach the transition state. So the difference in the energy between the reactants and the products here is termed as the delta G, the energy change. So okay. So activation energy is often supplied by the heat energy that the reactant molecule absorbs from the surrounding. The absorption of heat energy make the molecule move faster, accelerate the particle, so increase the rate of effective collision. So understand that it's not collide only, we want to collide at the correct orientation. Even though we know that heat can increase the rate of reactions, allowing reactant to attain the transition state, the question here is, it won't work well in biological system because high temperature will denature the proteins and also speed up unnecessary reaction. Means that, oh, no specificity. So therefore, what we need to do here is we have to use enzyme. The organism have to use the enzyme to increase the rates of reactions. Okay.